Hello, I'm Ned Talley, and today I'm going to be talking about the Brain Initiative Connectivity Across Scales program. This is 3 new funding announcements which launch the Brain Connects Network and mark the start of a new transformative project from the Brain Initiative, enabling unprecedented capability for brain-wide maps of neural circuits. If you're interested in learning more about this program and other Brain Initiative programs, I recommend that you go to braininitiative.nih.gov for lots of general information. This video has information for applicants to the funding announcements that provides an overview of program goals, application instructions, and project responsiveness. Yes, so there are 3 funding announcements. 2 of them are for comprehensive centers. We have a comprehensive center for human and non-human primate brain, and another announcement for comprehensive centers for mouse brain, and then a 3rd announcement for specialized projects. And I will describe the differences between the 2 in more detail below. Receipt dates are start this July, and then we'll have another receipt date in June of 23 and another one in 24 that's not shown here. For information, you can email us at brain-connects-inquiries@nih.gov, and we'll be happy to answer any questions. Okay, essential information. First, if you're an applicant, read the funding announcements carefully. So the announcements all have application instructions and review criteria that are aligned with each other and that are aligned to the announcement goals and the program goals. And these instructions will be the basis for peer reviewers and programmatic staff to evaluate your applications and make funding decisions. Another thing to know is that nothing that I'm saying here today or that is in these slides is supersedes or substitutes for anything that's in the FOA. It's really just a summary. All of the required information is contained in the FOAs. Now, many people have contacted us with ideas that are related to these programs, but might not fit so well. So if you are in that category, send us an email at the email that I show here, and we'll be happy to help you think about which program might be best for your project and how to fit it best. Certainly, there are lots of Brain Initiative funding opportunities with great funding lines right now, and there's other programs at NIH, and you are always welcome to submit an R01 to the parent announcement. So, overview of the program. So, this is a coordinated effort aimed at developing the research capacity and technical capabilities to generate and interpret wiring diagrams with goals of brain-wide coverage and comprehensive mapping. We anticipate that it will be a 10-year program. So this first 5 years of the program is focused on the innovations and the iterative engineering that are required to develop technologies to make this feasible. In other words, in 5 years, we want to be able to say that we can map the brain comprehensively and cover the whole brain. Now, we anticipate that there are complementary approaches to doing this, and that those approaches represent technologies that are suited for different types of research questions and use cases. And I'll be describing a little bit more detail below about the requirements that would enable the different approaches and technologies. As an outcome, we're expecting new capabilities and new discovery for mapping brains. And the kinds of discoveries we're looking for in the end are going to be new cell types, new circuit motifs, new wiring principles and circuit elements, and new computational and conceptual models based on the new kinds of data that will be possible. Okay, so the 2 types of projects, there's the comprehensive centers and the specialized projects. I'm going to start with the comprehensive centers. These are 5-year UM1 multi-component awards. There's a 30-page research strategy. Receipt dates are 2022 and 2023, and they're separated into human and non-human primate versus mouse. So for human and non-human primate, the requirement is for brain-wide coverage and comprehensive mapping of region to region connectivity at the level of axonal projections. But in the mouse, we have a different requirement, and that is brain-wide coverage and comprehensive mapping of local and long-range cell-to-cell connectivity at the level of synaptic connections. Now, it's useful probably to note that comprehensive could be a matter of interpretation here. And it does give applicants a bit of room for making the case. But remember that all of these instructions, again, are going to be given to the reviewers. Program staff are going to be looking at them. And so you're going to need to make the strongest case that you can for your approach. For the specialized centers, they're analogous to the comprehensive centers, but a full-scale pipeline is not required. And I'll describe that in more detail below. These are 3-year U01 single component awards with a 12-page research strategy. And we have 3 receipt dates, 2022, 23, and 24. And note that even though they are only 3 years and are intended to be smaller than the UM1s, the budgets are actually not limited. So if it requires multiple teams, you just need to justify the budget. And so that is something to consider when you're thinking about whether you want to try for a UM1 that's fully comprehensive and covers all aspects of developing this pipeline versus a specialized project. Okay, so I'm going to go into more detail on the comprehensive centers now. Okay, so the overall goals, as I said, research capability, research capacity and technical capabilities, brain-wide coverage, comprehensive mapping, and I told you already about the rules for human and non-human primate versus mouse. We're expecting that people will be collecting large subvolumes of the CNS that are sufficient to demonstrate the feasibility and to inform NIH on program continuation in a subsequent 5-year period. So really, we want enough data so that you show that these things are feasible, but really we also want you to just to develop the technologies and make it feasible. So the expectation is that it's complete pipeline. So you have to establish and scale up every aspect of this, going from sample collection all the way through data integration and dissemination. And the expectation is that you'll be at the cutting edge of state-of-art methods and developing that out. And we expect that you will apply the data to research use cases of high importance to the neuroscience community. So at the end of 5 years, not only do we want these capabilities, but we want new discoveries that will show why this kind of data collection is valuable. So comprehensive centers, now I'm going to go into the application instructions. The specific aims, be careful about what it says here. Summarize the overall goals and how each of the different components will be developed to demonstrate feasibility of scaling to entire brains. The limitation here is you want to be able to collect entire brains in a potential subsequent 5-year production period. And then the research strategy is 30 pages. The first 6 pages are center overview. That's relatively straightforward, so I'm not going to talk about it here. The research activities and milestones, that's 24 pages, and it's divided into 5 research activity elements. I'm going to be talking about each of them in detail below, but you have to start with the instruction here that says that for each of these elements, you need to explain the strategies and the innovations that will enable scaling to high throughput and that will serve as a platform for mapping entire brains and that will advance technologies and capabilities beyond current state of art. So you need to make that part of the entire application for each of the different elements. Okay, so the first element is sample processing and data acquisition. And the instructions are to provide details on tissue collection, processing, handling, as well as imaging and other data acquisition modalities. And the second bullet is key here. Explain how these processes will be automated and scaled for high throughput, including quality assurance and risk mitigation strategies. And then the third bullet is something also to keep in mind. You are required to focus for each of the different FOAs on either human or non-human primate or on the mouse. However, if you need to use a different species for limited testing and optimization, that's allowed. You need to justify it in terms of the scientific and cost benefits. So we don't expect that technology development necessarily needs to proceed using, say, a large brain, which could be prohibitive from a technical or cost standpoint. but instead other tissues can be used. Okay, the second research activity element is data processing and management. This includes data transfer, storage, and security. It also includes the data processing step, such as 3D registration and segmentation, and statistical representation. And you need to explain for each of these how performance will be assessed and optimized, and we're expecting cutting edge work, including new developments from the computer science world and the commercial IT industry. And we expect also that compatibility with existing resources really should be prioritized. So for automated analysis also, ground truth benchmarking is a major consideration. So if we're talking about automating, we do expect that you will be developing ways to develop ground truth data that are scalable, and we want to see cutting edge developments in that as well. In the end, what you need to do is explain how the approach is well suited for efficient and cost-effective data processing and analysis, and how it will enable data integration into a wider data ecosystem within the Brain Connects network and beyond. The third research activity element is integration and dissemination. And it's very key, it's very important that you take into consideration each of these three components that are bulleted below. So provide strategies or software tool sets for integrating new data of the same modality, integrating across modalities into the wider knowledge base, and dissemination to the research community. So data of the same modality, what we expect is you're going to be producing a large data set, but we need to see how new data will be incorporated into that. And that could be new data with more detail, or it could be comparative information from additional subjects or animals. In addition, we're expecting that you will develop ways or strategies or tools for integrating the data that you collect to other modalities. And the other modalities we're not specifying, but they could include molecular, morphological, connectivity, and/or functional data. What we want to see is that you have a way for reaching towards a unified understanding of brain cells, circuits, and networks. And then finally, the dissemination, you need to have a strategy, and this could include tool sets for making the data available for easy access to the research community, for collaborative proofreading and error correction, annotation, but most importantly for mining and discovery. Okay, the fourth activity element is research discovery. And this is where you get to show the importance of the data for a particular scientific question, to show how the data type that you're collecting is going to be leveraged. So for the chosen CNS volume, apply one or more key research use cases to demonstrate this value and the significance of the approach. And you want to try and apply it to a question for which understanding anatomical structure and connectivity will elucidate fundamental principles of neural function. Now, you are allowed to propose functional experiments to record and perturb circuit activity, but this has to be well justified, and it has to be limited in scope to complementing and further framing the significance of the corresponding connectivity data. So we really want to make sure that you have the opportunity to leverage the anatomical data that you're collecting to its fullest extent. And we are willing to fund functional experiments in that regard, but you do have to justify it. And this shouldn't be something where the functional data then is primary. Okay, finally, the final element is feasibility metrics and milestones. So what we want you to do is tell us what are the metrics by which your approach should be judged in order to demonstrate the feasibility that we need to assess. That we want to show that you can scale to entire brains, but we want to know what the measure is going to be that you have to hold yourself to and that we need to understand. And in addition, we want to be able to show how this compares to other approaches. So we need benchmarks for that. And they need to include expectations of the cost and the time and any ancillary requirements that may need to go into the consideration. And then finally, we are expecting in that 30 pages for you to have a timeline and annual milestones. And the milestones should use SMART criteria, meaning specific, measurable, attainable, relevant, and time-bound. And we would like to see a separate delineation of milestones that represent aspirational but realistic goals for the project versus go, no-go criteria that say that this project is successful and if you meet this minimal threshold. Okay, that was the comprehensive centers, and now I'm going to move on to the specialized projects. So the overall goals for the specialized projects are proposals to develop current or emerging technologies for comprehensive atlases of brain connectivity to enable faster, more precise, more cost-effective generation and interpretation of brain-wide wireless. So these are specialized projects, meaning they don't have to develop the entire pipeline. They can address any aspects of the pipeline, meaning they could be addressing data collection, reconstruction, analysis, integration, dissemination, and interpretation of brain connectivity and the associated data pipelines. The emphasis is on human, non-human primate, and mouse, similar to the UM1 comprehensive centers. But projects, specialized projects using other species are permitted, but they have to be well justified, and they have to be for approaches that can be generalized across species. We're encouraging people to propose specialized projects that would develop distinct capabilities that might not be ready to integrate into an entire UM1 comprehensive center. Just those distinct capabilities, if they're developed out, they could be incorporated at a later date. Okay, so for the application instructions for the specialized projects, specific aims, explain how the project will contribute to scalable pipelines for brain connectivity and associated data. The research strategy, once again, is 12 pages. Detail how the project will result in new and improved capabilities that can scale to whole brains, and advance state of art, address gaps and challenges, and offer advantages over alternative methods. Now, if you're generating a new data set or analyzing a new data set, you should explain how it will contribute to novel research findings and new capabilities. And if you are using species other than mouse, human, or non-human primate, you need to justify it. And then finally, we do expect a timeline and performance metrics to develop these, to demonstrate feasibility of scaling to entire brains and for comparison to other approaches. All of the FOAs have a couple of attachments. One is the resource sharing plan, which I think is straightforward. You can read about it in the FOA. The other is the plan for enhancing diverse perspectives. This is a relatively new development for the BRAIN initiative, and it's one that you need to pay attention to because applications lacking this PEDP attachment will be withdrawn without review. And the PEDP is evaluated by the peer review committee and is considered in the scorable criteria. So what we're expecting is a one-page summary of strategies to advance the scientific and technical merit of the project through expanded inclusivity. It should provide a holistic and integrated view of how enhancing diverse perspectives is viewed and supported throughout the application. It will be evaluated, as I said, according to the review criteria, and it can incorporate elements with relevance to any of these listed review criteria. Now, we have a lot of information on further guidance, frequently asked questions, and we provide key elements and examples if you go to the following website. This is on the BRAIN Initiative NIH.gov website. If you type in BRAIN Initiative PEDP into Google, this will come up. Okay, finally, non-responsive applications. This is really important. You need to follow the instructions of the FOA. And if you're not doing if you're not going after the kinds of things that are part of the program goals, you might be subject to having your application withdrawn. The non-responsive applications are projects that are not aimed at comprehensive pipelines for connectivity mapping, and that are technologies that are not expected to provide comprehensive mapping of region-to-region connectivity in the human or non-human primate and local and long-range synaptic connectivity in the mouse. And also technologies that are not expected to demonstrate the feasibility to scaling to Atlas's entire brains in a potential subsequent funding period. For the specialized projects is very similar. It's projects that are not aimed at those things. You don't have to have a full pipeline, but you have to be aiming for a component of the pipeline that I talked about it before. And then again, as I mentioned, any application lacking a PVP is non-responsive and will be withdrawn. But special review criteria, as I said before, the review criteria are very much aligned with the application instructions. And in fact, they're closely matched. For each of the bullets in the application instructions that I went through before, there's a corresponding question for reviewers asking them to evaluate whether or not you answered that bullet. So you will be evaluated according to whether or not you are going after the goals of the program and the specific application instructions. So the one thing that I really can not emphasize too much is read the announcements carefully. But we are happy to answer questions. You can contact us by email at brain-connects-inquiries@nih.gov, and we're happy to answer any questions either by email or we can set up a time to discuss. And with that, I will end the presentation and wish everyone good luck.